Hello, we are on the Pharmaceutical Drug Analysis book. This is a page 9. We are on the bioavailability. According to a biopharmaceutics expert, the term bioavailability may be defined as the rate and extent to which the ingredient is absorbed from the drug product into the body or to the site of action. It is measured by blood, serum, or plasma level or from urinary excretion data. Part 1. Importance. There are three major factors that govern the efficacy of a dosage form, namely onset of therapeutic activity, intensity of the therapeutic effect, and duration of the therapeutic effect. The above three factors are solely responsible for the rate of absorption of the drug. The distribution of a drug throughout the circulatory system and above all the elimination of the active principle from the body. Official quality control methods adopted, for example, disintegration time and the dissolution rate, do not give ample therapeutic equivalence among drug products belonging to the sample classes. Moreover, even the products of the same manufacturer may have varying degree of bioavailability in different batches. Therefore, it has become quite necessary to introduce comparative bioavailability studies and skillfully designed foolproof clinical tests of therapeutic equivalence as an effective true remedial measure of the ultimate performance of drug products. In 1968, 51 patients suffered from an epidemic of anticonvulsant intoxication in, Brisbane. A thorough investigation revealed that the intoxication was caused by altering one of the excipient from calcium phosphate to lactose in the drug product phenytoin capsule without adequate pre-testing by the manufacturer. This, excuse you, that my pet is also asleep. This apparent minor change of excipient was sufficient enough to bring about an appreciable major change in enhancing the bioavailability of the active principle to abnormally high levels in the affected patients. Question of quality. It has now been established beyond any reasonable doubt that quality of a drug products cannot simply be ensured by inspection or analysis, but a control system has to be built into from the very beginning of manufacture of a drug. Beside effective quality control measure, exercise in every aspect of production, including environment, screening of raw material, process control, intermediate shelf life of finished products. The most important aspect is to assess the bioavailability of the active principles. Difference in bioavailability, particularly in drugs with a low solubility, excuse me, with low solubility, as uh, as as ascertained by blood level attainment study appears to be caused by a number of formulation variables, namely particle size, crystalline structure, binding or disintegrating agent, excipient, etc. On the release pattern of the drug in its dosage form, for example, the rate of dissolution of the drug in tablet or a capsule in the gastrointestinal fluids. The third part, clinical efficacy of drugs. Medical scientists mainly rely on the measurement of bioavailability of a drug as a positive indicator of therapeutic equivalence. Because clinical efficacy for orally administered drugs depends on the degree of absorption and the presence of the active ingredient in the blood stream. Technical information based on in vivo standards and specification are generally incorporated in various official compendia. Hence, in order to record a legitimate assessment of bioavailability, in vivo test is an absolute necessity, and the relative data obtained therefore should form an integral part of the standard specification in the official standards. The fourth part, adverse drug reaction. Any dosage form can produce adverse drug reaction. Hence, a regular feedback of relevant information on such adverse reaction from the medical practitioner to the appropriate regulatory authority and the concerned manufacturer would not only help to intensify better safety measure but also widen the scope to improve drug design by meticulous research scientists all over the world. The following two examples convey the implication of adverse drug reaction. They are example 1, aspirin. Increased gastric damage and subsequent bleeding caused by some aspirin formulation have been specifically attributed to the slowly dissolving aspirin particles in the stomach. However, both effervescent and highly buffered dosage forms antacid aspirin tablets, which help in maintaining the aspirin in solution. However, both these in maintaining the aspirin in solution have been found to minimize gastrointestinal toxicity. Okay, example 2, chloramphenicol and tetracycline. Sparingly soluble broad spectrum antibiotic TB. Excuse me that my pets always bark. Sparingly soluble broad spectrum antibiotic like chloramphenicol and tetracycline found to damage the gastrointestinal epithelium besides changing the normal microflora in the GI tract that are required for normal good health. The fourth part, the next part, the identification test. Okay, I will record the identification test for the next video. Now we go to the bioavailability, and we are yet in the first chapters. Thank you so much for your attention. Please don't forget to like me, and my pets always sleepy, and pay attention to me. Thank you so much.